Thanks to all of you for being here. My name's Ed Lazowska. I'm from Computer Science and Engineering, and it's a great pleasure to introduce Jevin West. Jevin is a postdoc in the biology department, and I had known about websites like Eigenfactor in the past. I had no idea it came from the University of Washington, I have to say. And Jevin came over to talk a couple of months ago, and I was just blown away by the visualizations that he'd done with Carl Bergstrom and others in the biology department. So there's just unbelievably neat stuff in looking at interconnections, for example, that you can determine by mining enormous amounts of citation data and then visualizing it in clever ways. And it's really gotten a huge sort of national exposure through Eigenfactor and other websites. It's very neat stuff. So Jevin, thanks for being here.
All right. Well, thank you very much, Ed. I thank Ed for inviting me for this talk, but also because I talk a lot about networks and think a lot about networks. I see Ed as one of those big, huge, important hubs in these networks, and he's really connected me to a lot of people outside my neighborhood, across the country and even in other countries, but he's now introducing me to people just in my own neighborhood, not hundreds of miles or thousands of miles away, but just hundreds of steps from my office. So that's been great. There's definitely no lack of good work going on here. So I'm excited about some of the relationships that we're building with people around campus. So thank you, Ed, and thanks for coming out here and listening to some of the stuff that I'm really excited about. So one of the challenges I kind of gave myself last night as I was putting together the slides was to try something. I want to try to give the entire talk, or at least the important parts of the talk, in one slide. I want to give you kind of what the main message is, and if you want to leave after that, you can leave, but if you want to stay and listen to the superfluous details, I'll give you the other stuff. I better not do that. So, okay, so this is my attempt at distilling what I really want to talk about today. There's a lot of different things, but this, if you know this, you'd get an A on the exam. So, if I was to give you just two pieces of information, well, actually, there's three pieces of information that I'll focus in on. There are 10 different nodes or dots or whatever you want to think of these as. They could be humans in a population where the pink nodes represent girls and the blue nodes represent boys, and the links between them could be the relationships. And all I'm going to give you, so all you really know, the color of the nodes, you know how many links each node has, and then you know the source of those nodes. So one of the things that we've been thinking a lot about as biologists that think a lot about, of course, biological systems, but social systems, we want to be able to distinguish entities in large systems like this. So the way to do that, and this is kind of the whole message of basically the whole talk, once you embed this back into the entire population, whatever that population is, whether it's a population of species in an ecosystem or whether it's genes in a gene-to-gene network, when you put those back in, when you go from here to there, these little nodes, these nodes that were just up there before, they regain an identity. They have some sort of, they have distinguishing qualities. They have different effects on the entire system. So really what I'm going to talk to you about today is this sort of principle applied to one particular data set. We work on other data sets, of course, as well, but one data set we're really having a lot of fun working on is this enormous data that's now becoming available. The citation networks are just growing like mad. And of course, they've been around for a long time. People have been studying them for the last 50 years. But the reason why it's so cool now, I think, is that a lot of these proprietary boundaries that have been around this data that hasn't allowed the network itself to be put together is, a lot of those boundaries, those proprietary boundaries kind of made by publishers and other reasons are starting to dissolve. And we're starting to put these networks together at scales, that a lot of the forefathers of bibliometrics would have just, been glad to come back alive to play with. So today I'm going to talk, so really the whole message, it's just this idea of counting for the network. So I'm going to keep hammering that and tell you our approach to why we think that sort of thing's important. So a lot of this work comes from this project that Carl Bergstrom and I started several years ago called Eigenfactor. And when people have asked me before, what is Eigenfactor? Well, there's lots of Eigenfactor sorts of things. There's Eigenfactor, the algorithm, and there's lots of questions that we were trying to address when we were designing some of these algorithms. But one of the ones I'm going to focus on today that's pertinent to the talk today is how can we better evaluate the scholarly literature, looking at the full network, and what kind of things can we do, and how can we study this social process, this enterprise that we call science. And then there's the Eigenfactor, the project, and that has to do with all the visualizations and the economics of scholarly publishing. It has to do with the mapping of science that maybe some of you have seen that I'll briefly talk about today. It has to do with more of the philosophy as well. And that sort of question, one of the questions we address for the project, again, this is a general question, is how does the structure, how does the topology of these large systems, how is it that the way they're connected affects the functioning of the system? And also, how does it affect the individual nodes within these large network systems? These knowledge systems. So in this, the one thing I do want to say is this question typically gets overshadowed by the sort of more applied questions like this, because eigenvector kind of took off and is being used by, for example, librarians to make collection decisions, and sometimes we don't get to talk about as much of the basic questions. But I do want to emphasize that these are the sorts of things that we're kind of getting at from basic science questions. So I'm going to kind of break the talk into 4 different things. I'm just going to really kind of hit on a little bit why I think citation networks are really cool. I know a lot of people, they know what citation networks are. They have an idea that how they're connected and how they can be used, but I just kind of want to go over that again. Then I'll talk about more of the nitty-gritty details of eigenfactor, and then I'll get into sort of the navigation, some of the stuff that we're really getting excited about and seeing how it's showing us ourselves how we can use these tools for actually finding different areas or moving around in this landscape of science. And then I'll just end briefly with a little bit about scholarly feedback and how I really think that we're kind of at a ripe time where I really think that the sorts of research that's being done in this area, or just the idea that all this data is now being available to study, could actually affect, either through the metrics or the tools that are built, how we do science. And I think that's it's kind of an exciting reason to be working in this area right now, because the data is now becoming available. We now have some of the conceptual advances, and we have the computational power to do things that we couldn't do before that will allow us to really harness all this great information that we're accumulating. So I'll first start with citation networks. So, I think everyone here, if you've read a paper or written a paper, you know what I mean by a citation network. But what I want to emphasize is that it's not so much this process that we follow each day where we go to get a paper. For me, I actually a lot of times just go straight to the references and look for an important reference. But it's this academic convention that's been going on for centuries, where we've preserved these trails that have been laid down by scientists over time. And when you scale up to very, very large numbers, you can start to get an idea of the collective movement of ideas over this landscape of science, and you can start to understand where ideas are coming from, how fields break apart, how they come together, and you can also really get an idea where science could be going and where it's been. So this idea, this beautiful property of the citation network was first kind of written about. I mean, people had been thinking about it. It was first written by Jstor Corpus in 1965. He wrote this little essay in Science talking about how, there's this great property of the citation network. The nodes in this network, the articles and the edges between them being footnotes or citations, when you scale up, that gives you a great idea of kind of maybe the hot spots of science, or again, where science has gone and where it could be going. How can we use this sort of great data set that we're putting together that's been well preserved for a long time to find important things? At that time, they couldn't really take on the sorts of things that we're able to do now because, again, you have those proprietary boundaries. We didn't have the technology to digitize all this information. So he was thinking way ahead. So when I talk about citations today, really, very simply, I'm talking about either where the nodes are authors and they have a group of papers, and we deal with big data sets where they're author-level networks and the edges are citations. They can be journal-level networks. That's the type of network we've worked on most of the time, where the journals are a group of papers from year to year, and we look at how those citations are linked together, or how the journals are linked together. But when I think about these sorts of networks, I mean, there's also the article level, which are time-directed, and those have interesting theoretical implications for thinking when you're using those sorts of networks. But I really think that, and everyone in our lab, this is kind of one of the themes that we go by. We really think of scholarship as the flow of ideas. And networks really are, at least the networks I'm interested in, are all about the flow of information. So I see, a model system. This is a great model system for studying any sort of if you're interested in information flow on networks, it's a really nice system because it's readily available, like I mentioned, especially now, just even in the last 2 years. We have the computational power, and the data itself is very, very interesting. And also the links themselves are really are well defined. So you have something where, as biologists, we work with networks where we're not sure about what that link means. We're not exactly sure what those nodes mean, but for the most part, we understand what a citation network is. This is an example of taking a large amount of citation data and looking at the flows of ideas in one particular time period. And of course, you can take cross-sections and look at different data. But this particular, what you're looking at is a radial diagram of the top fields within science. This represents the 400 top journals and some of the top fields within the JCR. And what you're looking at there on the outer ring are the fields. So you see physics, you see chemistry. If you want to look at neuroscience, let's find computer science here. I know there it is. You can click on an entire field, and you can look at the flow of ideas. And actually, what we're doing here is we're summing the citations from a particular journal to another one, and also the citations that are going the other direction. So you can then, on the inner ring, you can actually look at the top journals. So since we're mainly a computer science audience, let's click on one of the journals here. And you can look at how, again, you're just looking, what this does is this isn't so much a useful tool for navigating, but it gives you kind of the sorts of things that we like to capture this flow, either through visualizations or through algorithms. And to get to here, of course, there's lots of steps. It doesn't just appear like this. There are algorithms underneath how you organize the data. But the idea is to come up with some way of trying to capture and try to see those flows. And there's all sorts of interesting stories that pop out. I mean, actually, I think the most interesting stories are the ones where it's not necessarily the connections that are existing or but actually the connections that don't exist. Like this is biochemistry. So why is it that they're not, why don't they talk to the field of economics? That's kind of, I mean, it's the fields that they're not talking to, I think, are just as interesting as the ones that they are talking to. So anyway, these are the sorts of things we start to play with and think about, and then we get back to really doing something useful. So what I'm going to show. Actually, one thing I do want to mention is, as everyone here who's worked with big data, scaling is obviously a challenge. And our first data set, for some people, I go, that's not that many. But our first data set was about 12,000 journals and about 150 million citations over about 15 years. And that's a pretty big data set. It's grown tremendously since then, and that's only in the last 3 or 4 years. But what happens when you scale up to these sorts of numbers, and you try to represent what I just showed, trying to capture flows or try to build the actual network? If you do that, you obviously get a black screen because there's more nodes than even there are pixels in the projector that we have today. So really, it kind of, the reason why I've shown this before, but really what it kind of does for me is it really hones in the sorts of questions that we're interested in. It's not so much about what information we keep, it's what information do we filter, what information do we compress. It's kind of the essence of the sort of statistical approach we have. How do we go from this messy big glob to something that has some sort of structure, some sort of semblance of what we would call a map of science? So we wanted to get to this, we started with the simplest question we could. We had this awesome data set, and we wanted to start with a very, very, very simple question, and that question was, how do we identify the important journals? I mean, even more generally, how do you identify just the important nodes in this network? And of course, there's been tons of work that's been done thinking about how you identify the important nodes in large networks. There's all sorts of centrality measures. But we had 2 caveats when we came up with this question. This was the sorts of, we kind of boxed ourselves in with this question. We only wanted to use the citation network. We didn't want to use any semantic data, at least for the first approach. But we most importantly wanted to include all of the citation network. We really think that when you think of these networks, there's a quote by my close colleague and friend Martin, who really thinks of these systems. He always said, a lot of people say, oh, the system is is greater than the sum of the parts. But he kind of always likes to argue against it. And he says, actually, the system is the parts plus the interactions. So really, if we think, intuitively, we think there's a wealth of information in these connections. And there's plenty of evidence and plenty of applications of other groups that have used the connections to say something about the system, then we want to make sure that we try to use all of the citation network. So that kind of led to the work in eigenfactor, so I'll kind of get to that. So briefly, for those that have never really thought about scholarly evaluation, and computer science is actually kind of interesting because you guys are kind of ahead of the curve a little bit in terms of kind of getting your ideas out faster. You have mechanisms, you have some scaffolding that's better than a lot of different fields in sharing of ideas, getting things published. Your sort of conference proceedings approach, there's some pros and cons to it, but I think for the most part there's mostly pros. But let me just kind of give you a general, 2-minute overview of what scholarly evaluation has been for the last century. It all kind of started with this particular paper that was written in Science by 2 chemists from Pomona College. They basically did what most of the field's been doing for the last century, which is just count stuff. So they wanted to figure out which journals do we keep on our collection. We're going to start a graduate program. Which journals do we keep so that we don't have enough money, we can't buy everything, so how do we do it? So they had this approach. They wrote in Science, they said, all we're going to do is we're going to take all of the citations that come out of the Journal of the American Chemical Society, which they considered the best journal at the time, and we're going to make a table and order which journals have the most citations, and that's how we're going to choose which journals we keep in their library. Pretty amazing. It turns out this particular table and this particular paper has had an enormous influence on the function and the way that we communicate as scientists, because librarians have used this approach for a long time, and this, of course, for those that know more of the history, led to the impact factor. And again, computer scientists don't worry as much about the impact factor as other fields. The life sciences really care about this. But for those that never really know what the impact factor is, it's a way of measuring the value, again, because we're trying to look like the very first slide I gave you, we're trying to figure out what are the nodes, how do you identify which ones are important and which ones aren't? How do you measure, come up with a measure of which nodes to keep and which not? So Garfield was interested in studying the frontiers of science, and he said, I can't keep all the journals if I'm going to study this, so here's my measure for keeping a journal. And all he did was simply say, okay, the impact factor is the number of sites. If you're looking for the impact factor in 2010 to articles in 2009 and 2008, and you divide by the number of articles, 2009, 2008. Pretty simple, right? Well, for those that have played any role in administration, or if you've been on a tenure and promotion committee, you know the impact factor has driven a lot of things in science and actually affected science itself. For example, again, if you've been on a hiring committee, you know that hiring decisions are based on this crazy simple metric. Library subscriptions are definitely we've been working with librarians for several years now, at least with our metric, and these sorts of things are used to make decisions. Promotion and tenure a lot of people that are on these committees will look to the impact factors. Ad placement, which is big money. Those journals that have a higher impact factor get more money for ad placement. This is the more controversial sort of approach now. The research assessment exercise in the UK is really looking a lot at, is using impact factor for their determining which universities get funding. University rankings are based on this. Basically, I just want to say that you look at something as simple as measuring something about the sociology of science, and before you know it, it's actually affecting the science itself. So this stuff matters. So based on this sort of theme about considering the source of those citations, impact factor, another way of looking at impact factor, if you were to take let's say you wanted to calculate the impact factor of plant physiology for the last 2 years, well, for 2010, you'd look at all the citations coming in from all these different journals, and let's pretend there was 3 citations coming from these different journals into the articles in 2008, 2009, so there was 2 articles, then the impact factor for that particular journal would be 1.5. But the thing is, with impact factor, it's a degree centrality measure, and it doesn't take into account the source of citations. And again, that's the sort of thing that we're trying to get at. How do you take into account the entire network? So going back to this picture, it turns out this is a real data set. For those that haven't seen it, this is a real high school, this is a data set from a Michigan high school where the girls are pink nodes and the blue nodes are the boys, and the links between them are relationships. And I'm using this just as an example of how you would ask the same sorts of question. Let's say if you were an epidemiologist and you wanted to see which nodes were most important for spreading, let's say, the cold virus or something similar. So it's really just a question of centrality. And again, I think a lot of people in here are familiar with the many different centrality approaches. But the 2 that I'll talk about is the degree centrality measure, which is basically impact factor, and eigenvector centrality, which is an eigenvector centrality measure. So if you were to do the degree centrality, you could take that same network, and all you do is just break apart all the links, you line it up, you say the number of students in each one of the categories. It turns out there's 1 boy in that network that has 9 links, and there's only 1 of those. and then there's 2 that have 5 links, and so on and so forth. Now you can see that all these right here, let's say in the category of 3, basically are all the same. You can't distinguish between these different nodes because, again, degree centrality doesn't take into account the source of the citation or the relationships. So another way of thinking about this, if you're accounting for the network, the impact factor, of course, is this sort of situation where it only just counts the number of links. And when you account for the network, this is eigenfactor. Well, this is the eigenfactor approach. And other people have thought about this, but this is what we're getting at. So, okay, so I've kind of spent enough time talking about that, and I could talk for a whole lecture about how the network does matter. There's plenty of examples. I mean, Google, of course, is the best example of how taking into account the structure of a large system can reap large benefits. But here's the interesting thing that I learned when I first started thinking about this problem in this interesting data set, that the network property of the scholarly literature was largely ignored for 100 years. There were some reasons for it, of course, because they hadn't really thought about it. But even till 2006 or 2005, really no one was still thinking about the network. Why not? So one of the goals of my research in general, it wasn't just for this sort of system, is how do we extract information from any sort of system so we can better measure this information flow? So there's a set of mathematics, and one of the things that I've come to realize, it doesn't matter what audience you have, showing how the mathematics works is always more effective than just going through it one step at a time. So just to remind you of what the challenge is. The challenge is we're going to play a game, and we're going to try to figure out which node is the most important node in that particular network. And I know there's different backgrounds here, so I'm just going to go and try to span everyone that I can. So in this particular network, you could look at it, and with at this sort of scale, you could start to get an idea pretty quickly which particular node is the most important. I mean, or you can make guesses. So what you might say, well, this particular node is, it's kind of central to the whole, if you're looking at the flow of information, if the links, just pretend, for example, these are journals, and the links between them the directed links are citations, and the thickness of the line represents maybe the number of citations going between nodes. Or you might say, okay, this one is, there's different ones that you could say, but as I kind of scroll over these different nodes, they correspond to a score over here. And there's two ways that I'm going to kind of explain what's going on. It's basically what I'm going to explain is the PageRank algorithm, but then show the nuances of what we do with eigenfactor on the scholarly literature, so we get kind of at the same thing. But what we're trying to do is capture all that information in those links. So let's play a game. So let's initiate the votes, and let's start everyone. It doesn't actually matter how you start this little game, but what we're going to do is each individual node is going to vote. And if you have, let's say, 3 links out, you distribute your vote equally amongst those 3 links. If you have 2, you do it through 2, and then we iterate the process. So each one of these nodes right now has the same amount of voting power, or let's just say currency or something. And then we go ahead and vote and see what happens. So as you vote, you start to see immediately that some of the nodes accumulate more of the votes because they had more links coming in. And if they accumulate more nodes or more weight in the next iteration of voting, then they're going to distribute more weight to what they're connected to. And then you allow this process to go over and over and over. And there are certain things that immediately a lot of people will notice. They'll say, well, look at this particular node up here. Nothing's connecting to it. So how is it accumulating any vote? Well, as many of you who have studied PageRank know, there are different correction steps to make this sort of system work or make this approach work. But the way that we do it, just like, again, the typical approach, is that each time you vote, something that you can't see is that there's just a small amount of your vote that goes to all the other nodes in the system. It's so you guarantee certain properties about this particular network, so you can actually do some of the neat things that you can do on these networks. So as you see, that goes really slow, but if you increase this, or actually just the speed, and watch this go for a very long time, you'll notice that this is going to converge to the stationary distribution here. And again, it doesn't matter how you start, but it turns out it doesn't take that many sort of iterations to get here. And again, this is kind of like PageRank. But where this kind of differs, if you let this go for a very long time, one of the things we realize for at least one of the differences in the eigenfactor algorithm is that at the very end, after it does converge, then we take an extra step. Because again, I explained how this particular node accumulates some vote, but really, do we really want this particular node having any sort of vote at the end? So what we do is we take an extra step. Turns out it seems like a very simple extra step in your algorithm, but it's incredibly important. We've done quite a bit of work looking at what this effect does when you look at rankings and how you compare things, and it turns out that that's actually quite important. So that's the deterministic version of how to interpret the algorithm. Let me show you also the random walk interpretation. It's an interpretation, actually, of the two. I particularly like this one, but if I reset, what's cool is actually the stochastic interpretation converges to the exact same answer that the deterministic version does. But basically the way to think of this one is that you allow a random walker to jump, let's say jump into the library and start randomly moving around the journal network. Whoops, let's just let it go. And you let this random walker move around the network, or you could actually think of it as an infinite number of random walkers on the network, and you let these random walkers go to work following these trails that have been laid down over time, and you find out after this long walk, where did the random walker spend, or where did the random walker, spend most of its time during this random walk. And again, there's the teleport correction that you see, this red dot sometimes jump. Every once in a while, let's say on average like every 6 steps, the random walker decides to just teleport. It turns out it's not uniformly in our particular algorithm. We do it based on the size of the journals. But anyway, it does this sort of correction. So you're starting to get an idea, at least, how do we evaluate the individual nodes. And then this kind of led to some of the work on the mapping stuff. But Carl is actually going to give a talk in the same room, same department, in about a month. And I'm going to let him get into the nitty-gritty details of the map equation. But I will show you some of the results of that. So I'll stop this. So this is just to kind of give you an idea of how the algorithm works. So, and this was an applet that was built by Daniel Elder, who was a master's student with Martin. Well, I've spent some time with Sweden, and I'll be going there again. And he's super, super good at doing these sorts of things. And we have all these new ideas of what we can do too. So, and hopefully, actually, there's one other new thing I might show at the end if I have time. But one of the things, we put up a website, and we thought, well, we kind of have all these rankings. Let's let people just play with them for free. Like everything we've built, we put up that's freely available. And you can see this sort of stuff. And also we have pricing information. That's a totally different story, but it's a very interesting story. The economics of scholarly publishing, just as much as the metrics themselves, it's a very interesting story. I'm very interested in this sort of thing as well. But also the eigenfactor metrics are included in the JCR, the journal citation port that Thomson Reuters puts out. And you can go there and find those rankings. I'm not going to really show a bunch of results because you can go there. I'd rather just kind of show maybe one result that shows these sorts of rankings over time. So going back, this is just one of many examples of the sort of data that you'll see. So the way to interpret this, many of you probably seen these sorts of graphs, but you're going to look at these rankings over time to see how they change. I think looking over time, of course, gives you a better idea of really what's going on in a particular subfield or for a particular journal, or it doesn't have to be journals. It could be authors. We've done author-level ranking, of course, and also paper-level ranking. But what I'm going to show you here is just kind of an interesting story. The way to interpret this, the size of the circle here is that eigenfactor. So that's that algorithm that we went through just a few minutes ago. And the color is actually the publisher. So blue represents Cell Press, which in the life sciences was typically the largest, most important publisher for a long time. And then the green dot, or the green circles, is Nature publishing, along with the yellow too. So yellow and green are both Nature. The only difference between yellow and green is that the yellow journals have the word Nature in it. That's the only difference. I'm not telling you anything else about the field or what the journal publishes or anything else. All I'm going to do is just show you that the word Nature exists or is in the title of all the yellow journals, and green is Nature-owned journals without the word Nature. So on the y-axis, you got article influence, which is basically the eigenfactor divided by the number of articles in a journal over a given time. So it's basically it's the eigenfactor normalized by the size of the journal. And the x-axis is the total articles. So let's go ahead and watch over time. This starts in 1996, and we'll watch it over the last decade. This is one of all sorts of interesting stories about how this, we think we're doing science independently, but it turns out that as a collective, we're certainly affected by certain little nuances and metrics and all those sorts of things. But it turns out, as you can see, having the word Nature is a nice strategy for getting a journal that does really well. Anyway, so that's just some of it. So one of the first questions you ask if you come up with some other way of measuring something, is how does it do? Does it differ actually from the traditional metrics? And it turns out there's quite significant differences. There are some similarities, of course, but there are differences. So all you're looking at here is impact vector, which is a degree centrality. Just think of it as degree centrality, because it's not just citation networks that we've done this sort of work on, but all you're seeing is the journal, the ordinal ranking on one side. Don't worry about the titles or anything. All I want you to see is there's lots of movement in the ordinal rankings. But what's interesting is this particular field, if you just look at it in aggregate, the correlation between these two metrics is quite high. So people say, well, it's 0.8 r squared of 0.82, so really is there any difference going on? And the answer is there is quite a bit. So actually one way of explaining it is to go back to the simulation. It's better even than just showing it in graphs. So if I go back to here, I can pull up an example network where is it here? There we go. So this is just a small network so we can get our brain around what is, so I've been claiming here for the last 20 minutes or whatever that degree centrality is actually quite, actually eigenvector centrality sort of measures are a useful way of capturing information about networks, at least using just the structure. So let's get an example network here. If you were using just a degree centrality measure, all of these, let's just color them the same, because actually the score for all of these would be green. So I'm just going to color the score based on what they would be. So a degree centrality measure, they'd all be the same, because this particular node up there, actually they wanted on TV for me to show you this. So the node here has one link coming in. This one has one link. This has one link. This has one link. And this has one link. So they'd all have the same score based on a degree centrality measure. But if you let this go, what you notice pretty quickly is that the size changes really fast. And that if you use an eigenvector centrality measure, those nodes in the network there's ways of distinguishing them. They're different. Now, depending on what you're measuring, that may not be what you want, but for the stuff that we're interested in, it is what we want. This is where the actual source itself is differently. So again, all these nodes are actually the same score based on degree centrality. This one is the same as this one, and this one. They're all the same. They all have the same score based on just counting citations, and if you want to divide by the number of something, that's basically you're just counting the number of edges coming in. If you do it by this approach where you count, then you start to see differences. Let's just let it just kind of go, and you start to see immediately. So by doing these little toy examples, we found it's really a powerful way of getting an intuition as to how these algorithms work. You can do the mathematics, that's one approach, and we do that, but this is another way of explaining the algorithm, but also understanding what's going on. All right, so the point is, all you have to know there is the two methods do different. So now there was 4 sections. I started with citation networks. We talked a little bit about eigenfactor and sort of ways that it's used. It's used by scholars and librarians now as an alternative. There's no best answer to any of these metrics. The best way is, of course, to read the papers. But it's actually one of the things that people are starting to use. It's become an industry standard in some realms of academia. And now I'm going to move to navigation. So navigation's kind of cool. There's all sorts of different aspects of navigation. So the question at the beginning of basically the first part of the talk, and also the part where we just the first part of the whole project itself, was answering this question: how can we better evaluate the scholarly literature, based on some of these philosophies and these approaches? We've kind of moved now to a similar question, but a little bit different question. We've gravitated to more navigating the scholarly literature. How do we use these tools to then better find papers, or to, just for our own good, when we're trying to look for something, when we're moving into a new field, if you like doing interdisciplinary research, how can you get the lay of the land so you can get going fast? So, in other words, we've thought a lot about ranking. Now we've kind of moved towards mapping, and how can we use these maps to better navigate. So what you're seeing here, I'm going to let Carl get into the details in a couple weeks about how we build these maps. But basically, the way to interpret these is the arrows represent the flow of ideas, or basically the flow of ideas between fields, and the size of the node represents again this based on this a little bit based on this algorithm that I've been showing you, where you have it represents the number, basically the number of visits to those particular nodes. And then also when you look at kind of the backbone of these, when you're looking at any of these labels here, this particular map we did on probably, I think this one was about 6,000 or 7,000 journals and about, I think it was about 20 million citations. But you can see kind of the major fields, of course, general medicine, molecular biology, chemistry, and you can see how all these different fields are connected. So there's been a lot of maps. Our approach, like again, once Carl gets into it, it's really neat stuff. But these are the sorts of maps, it's this approach that we use to do some of these navigation things. So we can use these maps for discovery, but we can also use these maps to discover patterns as well. So one of the first things we did when we got these maps, we wanted to use them for something. So one of the things that we did was we took this map of science, and then we looked at all the journals within the different fields, and we looked at their impact factor. And we wanted to see, we have this sort of heat map of all the different fields. The darker, the purple or whatever, actually I don't see colors very well. The darker color there is the higher the impact factor in that particular field. So there's all sorts of ways of trying to figure out why is molecular and cellular biology have typically journals with much higher impact factor. And again, like I said earlier on in the talk, impact factor basically equals money. So these sorts of things do matter. So it turns out, I can make just a long kind of story short, that the impact factor, this gradient that you're seeing, an impact factor across this particular map of science, is being driven mainly by the coverage. What we mean by coverage is the number of citations that are going into the corpus that we're analyzing. In this case, it's the journal citation reports. So you can see that these, and we've done the modeling and stuff, but you can actually see that this particular map here maps pretty closely to just by looking at it. I mean, we've gone through and done this a lot more rigorously than just looking at two different maps. But this gives you an idea. By looking at these maps, you can actually do something useful with these maps quickly, because we can see patterns. Of course, we have this high bandwidth for seeing patterns, so these are the sorts of things we like to do.
The size of the node is numbers of papers?
Nope. In this case, it's the same map before. The size of the node is the number. It's basically the traffic going into that particular field. So it's in that case, if you were doing this random walk, all the particular journals in that field, there'd be a lot of visitations to that particular field, molecular and cell biology. Okay. And that's just because of the representation in that. Well, it's not always, but the JCR has a lot more molecular and cell biology and computer science. As you can see, actually, computer science is kind of tiny. You probably can't see it very well, but it's way up there. But they don't have very many computer science nodes. Yes?
What's the impact of the number of people working in a field?
Yeah, so that actually has an impact too. One way you could do this different things. You could just look at the number of papers, and you could see how many, does the number of papers within the field affect this inflation in, well, we're not even looking over time, but does it affect this, does this have an effect on this? And of course, the number of people working in the field does affect. There's all these different things that have effect. The one that had the most effect, again, is this coverage, the number of citations coming into the JCR. But it does have an impact for sure. And in this case, if you added up all the people working in molecular and cell biology, it'd be far more than computer science, because it's just not a very good representation. Another thing you can do, one of the things that we were interested in, we built all these maps, but we wanted to see, how is it that, what are the differences between these two? You can build a map in 1995 and 2004, but what's really interesting is to try to figure out what are the differences between these two maps. So one approach is, oh yeah.
So I don't get it, because for example, you've changed the labels on the node.
Yeah, exactly.
Automatic or what?
Oh yeah. So that's a really good question. So the labels themselves on these particular nodes. So the mapping algorithm, which I feel bad because I'm not getting into details about how the map equation works to get these figures. That's what I got to give Carl something to talk about. So no, just kidding. Carl has a lot more to talk about than I do. But this particular map is. The labels on this particular map are done by hand. So they're clustered, so we know what the cluster is. So in this particular cluster in general medicine, we see after we get this output, let's say 3,000 journals. And we see immediately that they're all general medicine journals. And so we give the label. But what I'm going to talk about further down is that we can actually automate these labels by looking at the full text. So in this case, it's not looking at...
So the big change here, for example, is there's that heavy arc on the right. That's the upper right-hand side of the triangle that just doesn't exist 10 years before.
Exactly. Okay. And you can go in these in detail. And actually, you don't need any tool. You don't need any sort of tool that I'm going to talk about to just go through and look at it. What we wanted to do was try to figure out a tool that does it kind of for you, does all the hard work for you, so you can kind of upload the hard work to the algorithm. But yeah, so what you're seeing exactly, Ed, is there is a big difference in these particular maps. And the labels is actually At first, I mean, that's the only part that's done with us by going through, looking at what's in this cluster and saying, well, this is molecular biology. But like I said, later on, I'll show you a little bit how we're trying to do that automatically. So these alluvial diagrams here this is something that Carl Bergstrom and Martin Roswall came up with. This technique for taking these different networks. So if you go back and you look at these particular, let's just pick molecular biology, for example, and go to here in 2001. Molecular and cell biology is this block right here. So that represents, it's that circle. So you basically take the map and you transform it into this vertical column. And in the column, each one of the blocks are represented by an individual field. Now, we're not showing all of them in this particular diagram. We're just kind of highlighting one particular thing that came about. But this, if you see, the way to read these is we did one of the maps. We transform it into this vertical column, 2001. You do it for 2003, 2005, 2007. And then you let the algorithm go to work. And again, that's what Carl's going to talk about. But just kind of give you a little taste of one of the stories that emerges is that in 2001 and before, and for a long time, neuroscience has been this standalone field. There's been neuroscientists that publish primarily, let's say, in neurology, some publish in psychiatry, some in medicine and molecular and cell biology. But what you see over time is that there is this conversion into one standalone field. And in 2007, you see it kind of on its own. When you look at the journals themselves, you can look at the data and see that these are the standalone fields. And there's obviously a lot more than this particular example. And actually, Martin's developed this tool that you can do on any data, not just citation data. You can do it on any data where you have networks over time. And you can look at these things and see what's changing and what's not changing. So one of the data sets I actually want to really get into seeing interesting things with the alluvial, you need more than just 10 years. So one of the data sets that we're working on that I think is really cool is the Jstor Corpus. And what we'd been working on before, and this was still a huge data set, we had 150 million citations over more than a decade, and we were looking at journal level data. We wanted to move to article level data, but we were still happy working in this little realm. But what's cool about Jstor is they have a lot more time. They have papers all the way back to Newton. And these papers all the way back to Newton allow us to really, because if these alluvials are going to tell us anything interesting about fields before even neuroscience was thought of, it would be really neat to have a data set to do it. And we can do that with Jstor. And then the other really cool aspect of this Jstor data is that we have full text. We've kind of avoided full text for a long time, but we decided, all right, let's get our feet wet and let's do a little bit of semantic analysis. But this is the sort of data set we're working with. And I'll show you some of the things that we're doing. So one of the challenges, of course, is using the map equation. Carl and Martin had figured out a nice way of partitioning, hierarchically partitioning these large data sets. And so you have a situation where you run the algorithm and you can get this hierarchical partitioning where you have science and then science breaks down to, let's say, biology and economics. Biology breaks down to these further fields. These aren't papers, they're actually fields, so there really shouldn't be arrows there. But these are individual fields. So one of the things I was interested in, Martin and Carl had kind of solved how to do this for journal level networks, but I was really interested in looking at acyclic digraphs of these particular, basically being able to look at time-directed networks where you could actually throw in an article level, big, huge article level network and get this partitioning. And the reason why that's kind of challenging is the time-directedness makes it a little bit more difficult. So what we have is a partition. So what we're able to do is we get this hierarchical partitioning, and then we have this challenge that you were bringing up, Ed. How do you get the labels? So what we've now done is using the full text, we break them into trigrams, and we put them in this big data set and assign all the trigrams to the papers into the clusters where those papers reside. And then we have this is work that's been done by Daryl in our lab, and you're able to then identify the labels, or you're able to put labels to these clusters. So you can use this as a way of navigating. So what we have, one of the results that we got from doing this a large amount of work, you get this clustering. And this is just an example of one of the clusterings. I mean, you get this for all clusters, and you get thousands of clusters. I should mention, I didn't make this clear. I'm actually not making this clear at all. But basically, this is a really hard thing to do, because you have a huge corpus of about 8 million papers, And actually, we're working on clusters that have 150 million citations, or nodes, and even more, hundreds of millions of citations. But we want to do this automatically, and we want to do it fast. Basically, we're taking a large data set, and we're trying to find ways of organizing it just by looking at the citations. And that's a challenge, and that's been a challenge for a lot of people working in the field. We still haven't completely solved it, but the thing that I'm excited about is that we've started to solve the problem of article-level networks, time-directed networks. By doing this, we can then start to understand what the difference is between citation flow groupings and groupings based on semantics. So let's say we get a grouping here, and this actually is a grouping where these ecology and evolution are more related, like molecular biology and ecology and evolution are more related than the social sciences. Those break up, and that's by looking at citation information, just looking at the links and the footnotes. Then you take jargon and you find interesting things like this. This is an actual result where if you look at jargon, it turns out that ecology and evolution can talk to each other better, and social science can talk to each other better than molecular biology. So just by kind of getting our feet wet, and this is all just really basic approaches that have been around for a long time on how to group things based on jargon, we get these interesting results. So you can do this for all fields. And okay. And so this map here represents groupings of all these different fields. The text is kind of hard to read, but what you're seeing here is, you have a lot of the social sciences over here, you have the life sciences, you have kind of molecular biology here. And basically what this map shows, I'm not going to get into detail on this because I don't have time, but what you're looking at, this was a map created by Daryl, this particular map shows which groups or which fields, which clusters within this large data set communicate frequently. Because the placement, the spatial placement for each one of these groups is based on citations, but the hills, the actual mountains that you can see in this little topographical map tells you which fields communicate inefficiently. So it puts things together based on frequency of communication through citations, but then there's a measurement. We look at basically the jargon barriers between different fields. So the other thing, this is all kind of in service of trying to understand the sociology of this thing we call the science enterprise. How is it that we can look at communication, like, for example, jargon barriers, or maybe communication inequalities? One of the things that we're kind of interested in is looking at these partitioning. So I've been talking a lot about how we can do this automatically. We can take this enormous data set. We can partition it. So this is all of Jstor. So this block kind of represents all of Jstor. This particular block is a subfield of Jstor, which is ecology and evolution. In ecology and evolution, you see plant ecology and all the other fields that are associated with that. Now we can actually move into these different fields, and we can start to zoom in and zoom out of these different fields. We can zoom back, and we can look, we didn't want to look at ecology and evolution. Let's look at economics, for example. The colors in this particular one, we were interested in looking at gender bias. We wanted to see, what's the represent. This is something one of the first easy things you could do if you wanted to look at the sociology of science. You could start to look at what the representation of women and men is in science. Now, as you can see, it's mostly men in economics, as you would expect. But down here, you can also then look at author order as well, and you can kind of get an idea of, are women actually being, kind of shafted in the first author position, or are they getting, sometimes they're above what they, based on the proportion in that field. So this particular tool that we have is giving us, we're actually working with people in gender research that are using this for the first time to be able to see over a long period of time, because we can break these into different time periods. And you can see that, like, if you move to 1990 and just 2009, you actually see, like, let's go to sociology. You see what women are working with. For those that can't see the colors very well, this is mostly red up in here. This is, a lot of women in this particular field tend to work on stress and coping, and the men work on crime and sentencing, I guess. It's kind of an interesting, there's all sorts of interesting stories in there. But we can zoom in and zoom out of these things, and we can really start to get an idea of what the representation, it's not just gender we're interested in, but just building these sorts of structures of data quickly and able to move and navigate. Again,
Are these labels automatically generated or not?
so the end labels that you get on here are actually done through the algorithm. So the ones that are up are the ones that we're having a challenge with. So it's talking about challenges. We're not good at this part, but we're really good at the end parts.
Cool. Great.
We wanted to see, and actually a lot of people have been concerned about this, that men are grouping with men more often based on the proportions of men and women. Basically, are we seeing this situation where men kind of author with men and women author with women? And we can actually answer that question now, and I can give you that answer. It turns out it's quite interesting. If we look at, we call it, it's basically F-stats for anyone that knows about F-stats. This is the entire corpus of Jstor. And the brown is more homophilous. And a lot of papers have been written recently that say science, and particularly many fields, show this homophilous, or show homophily, which is men tend to write more with men, women tend to write with women, and this is part of the explanation to the inequality that we're seeing in science with women. Turns out, it's not that simple. If you then look at the entire structure of science, and the only way you could do this is to have this hierarchical partitioning, this map, this tool that I keep saying is useful, and here's one way of finding just one of several examples. You can actually look at the entire partition at once. Actually, let's just look at one. Let's look at molecular and cellular biology. What you find when you take structure into account, you start to see that, if I let all go, that when you take into account the proportions in each one of the individual fields, you see what's called the Walland effect. It's something we see in biology, population genetics, so much that we have a name for this effect. When you take into account the population structure, you see a much different result. It's also referred to as the Simpson's paradox, for anyone that knows about the Simpson's paradox. So there's all sorts of interesting things. I mean, if I had more time, I'd love to get more into those stories. But I just want to end with this last thing here, that if you really want to talk about discovery and using these tools for discovery and navigation, like I keep hitting, you need kind of a compass. And so we developed, based on a lot of these data sets, we've been working with Microsoft Research, Jstor, SSRN, Thomson Reuters, ADIA, a whole bunch of different data providers, and we built a recommendation system based on this map equation. And Carl's going to talk more about that, but I will show you just kind of how it works. So since you guys are computer scientists, I decided to choose the computer science corpus. And the idea behind this recommendation system is that you can put in any author, let's say Kleinberg, just kind of have this preset. It orders all of the papers in all these corpus, or all these corpora, I guess that's the right word, and you can click on an individual paper, and it'll come up with 4 recommendations. Again, this is kind of, we think of this as you can use the maps themselves, and then you need a compass. So there's an expert recommendation system, or expert recommendation. That's for someone that knows the field really well, and if you've read that, you kind of know the field, you want something that's like the key paper. That might not be the one you want if you're a graduate student or someone new to the field. You'd need a classic paper. So we have this classic recommendation, and then we have we call it the hot recommendation, but it takes into account the time. So it'll look at the hot paper in that area, looking at these accumulations of citations over time. And then you have this serendipity, which is another thing. But you can use these sorts of things. We're building an interface where you can use this recommendation system next to this map. And this map actually represents about 7 million citations just in computer science. And again, the labeling is a whole challenge, which I'd like to talk to the computer science department about it. But you can use these maps and get an idea of how this thing maps together in a particular time period, or, like we're trying to get to, over time. So let's say you're interested in operational, well, let's go to discrete mathematics. If you're interested in that area, you can double-click on discrete mathematics, and you'll be able to look at the top journals, or you can actually put top articles or top authors or whatever you want to be able to move in and out and at the same time have these recommendations. So it's being able to use these different tools for moving around science. So I'm going to basically end there. I don't have time to talk about this, but basically it's this idea that scientists will react to these metrics. And there's some important issues here that I'd like to address. But before I end, I might as well conclude with everything, just with 5 things. What's the question? We're interested in information flow in social and biological systems, and we need basically a model system. We think the scholar literature is a really good model system. Some of the challenges we have right now, which I'd love to talk to you guys about it, is the semantics and some of the data viz, of course, and just dealing with big data in general. This isn't our area of expertise. We'd love to get feedback. The concerns are this feedback, but I didn't really talk about it. And then the relevance is I really think that we're at a time that if we do this right, I think we might be thought of as the generation that was able to organize the information the best, and I think it's an exciting thing to be a part of. So I want to, of course, thank Daryl, who's been a huge part of all the stuff we're doing. He's actually going kayaking right there. Martin, who's actually skiing. As you can tell, our lab doesn't do anything but play outside, but that's Martin. He's done a lot of the work on the map equation, and Carl basically does everything. And actually, just to plug again, I've said it like 4 times, but come to Carl's talk in January. It's going to be really good. So anyway, there's lots of other people to thank. I can't even list all of them. It's a really fun project. I kind of just gave you a smattering of some of the things. Probably wasn't quite clear on some things, but please come ask me, and I'll make it much clearer if you come talk to me or come over. I'm actually just across the campus. I'd love to work with anyone who's interested in this sort of thing. So anyway.
Thank you.